Okay, so this is chapter 5, the structure and function of the plasma membrane, and this is part 1. Okay, so we've gone through all of the organelles found inside of a cell, and we've actually already talked about the plasma membrane, but this chapter really focuses on that structure. The plasma membrane is also known as the cell membrane. Those two terms can be used interchangeably. They mean the same thing. The plasma membrane is going to define the border of the cell. It separates the inside of the cell from the outside of the cell. There are also internal membranes that create compartments. We talked about those in chapter 4. So for example, the nucleus is surrounded by a double membrane called the nuclear envelope. Just outside of that, really connected to that, is the endoplasmic reticulum, which is a series of membranous vesicles and sacs. The Golgi apparatus, same idea, membranous structures. The mitochondria, double membrane structure. Chloroplasts, if we're talking about plant cells, double membrane structure. So there will be internal membranes that create compartmentalization, and then at the exterior of the cell we have the plasma membrane. And remember that plants also have a cell wall, animal cells also have an extracellular matrix, but again our focus is going to be on the plasma membrane being that border of the cell. You are going to hear me say over and over again that the membrane is selectively permeable. And we'll talk about what that means, but just to give you an idea, that's going to mean that only some substances are able to cross freely without the help of a membrane protein. Okay? And when we say selectively permeable, that could be crossing from outside of the cell to the inside or from the inside of the cell to the outside. So it could be both uptake as well as excretion. We say that membranes are very flexible, and we're going to talk about this fluid mosaic model, right? Flexibility. Membranes can kind of change shape. They are self-sealing. So if there's a disruption in the membrane and those phospholipids come apart, they go right back together. Okay. The membrane also participates in some other activities that I'll mention. I'll mention it again a few times this chapter, but we don't really get into too much detail. But this point down here, the membrane does carry markers that allow the cell to interact with the cell's environment. So for example, it allows cells to adhere to each other, to stick together, or to adhere to a surface, or allows cells to actually move around, okay, if there's a cell that moves from one part of the body to another. There will also be markers on the cell's surface. These will be carbohydrates, and I'll point this out a little bit later, but they're going to allow your immune system to recognize your cells as belonging to you, which we call self, versus anything foreign that makes it into your body would be non-self. Those cells would not have the same surface markers as your cells have. Okay, and lastly, the membrane is responsible for what we call signal transduction. So the membrane is going to have protein receptors in it, which will bind to signal molecules, and then those signals will be transmitted to the interior of the cell. So the plasma membrane is really important. Again, its major role is to define the border of the cell and to make sure that the cell maintains homeostasis. So it is that outermost border that's going to ensure that. We'll do just a little history of how the plasma membrane structure and components were figured out. So the plasma membrane was actually identified way back in the 1890s. And actually there's some evidence that that date might even be further into the past. In about 1915, scientists started to analyze membrane components. And again, there was multiple labs that kind of get credit for this depending on what you read. And so what was found is that membranes are composed of two major components. And there's some other components as well that we'll talk about, but really two major components were found. And they were proteins and lipids. And it didn't matter what type of membrane was analyzed, they found these same two molecules. So proteins and lipids. Calculations were done that compared the total number of lipid molecules to cell surface area. And what scientists figured out is that the membrane could be no more than 2 phospholipid molecules thick. And so you've seen this picture already, right? In the last chapter, you can see that this is a lipid bilayer. So we've got one layer of lipids, and here's my second layer of phospholipids. Okay, and so again, way back in 1915, scientists kind of figured this out. Membranes are composed of proteins and lipids must be in a bilayer. Okay. So a couple of scientists that get credit for this model, which is actually not true, but it was accepted for a long period of time, and so it makes its way into science textbooks. So 2 scientists by the name of Davison and Danieli in 1935, they came up with what was called the sandwich model for the plasma membrane. And they said that yes, the plasma membrane is made up of a phospholipid bilayer, so 2 layers of phospholipids. We already kind of knew that. And that layer of phospholipids is sandwiched in between 2 layers of flat proteins. Okay? And so that's what this model looks like down here. So you can see the phospholipid layer here and the phospholipid layer here. And then these purple kind of flattened structures are the proteins. And so again, the bilayer is sandwiched in between 2 layers of proteins. In the 1950s, when electron microscopy started to produce good imaging, transmission electron micrographs, there's one shown here, kind of supported this model. And so you can see it looks like a railroad track, right? And that's how it's been described. This is the plasma membrane. This would be the cytoplasm of the cell. And this would be the extracellular fluid, so outside the cell. And so this looks like, sure, maybe the sandwich model is correct. In fact, that model was accepted until the late 1960s. And so it was accepted for many years. Okay, and then in the late 1960s, scientists started to question that model. Some things that made them question that model was, one model for all membranes doesn't work. All membranes aren't going to be identical to each other. The other thing that scientists were figuring out is that proteins, just like phospholipids, are amphipathic. You've seen that term already, right? Let's make sure we understand what that term means. So amphipathic means that proteins are going to have hydrophilic and hydrophobic regions. And so it doesn't make sense that all the proteins would sit outside the cell in the hydrophilic, watery environment. Some parts of them must be tucked into the membrane, into that hydrophobic core. And lastly, not all proteins are flat. They come in a variety of shapes and sizes. And so one model, again, for all membranes doesn't work. So in 1972, 2 scientists, Singer and Nicholson, came up with what is called the fluid mosaic model. Okay, and that's the model that's still accepted today. Lots of evidence to support this model. And the model is depicted here kind of in a simple way. And on the next slide, I have a more complex model to show you. And so you can see here the phospholipids, right? So here is the hydrophobic head of this phospholipid here. Sorry, hydrophilic head, right? The polar head group. And here's those hydrophobic tails. We'll look at this molecule in detail again. It's in a bilayer, so we have 2 layers of those phospholipids, and then proteins are shown in purple. And so this protein is extending all the way through the membrane. We're going to name that a transmembrane protein, an integral transmembrane protein. The hydrophobic core of this protein is, buried in the hydrophobic core of the phospholipid bilayer. The hydrophilic regions get to sit outside, right alongside those polar head groups. This peripheral protein, right here, would also be hydrophilic, okay, or at least the outside amino acids would be. Okay, and so what Singer and Nicholson said was that membrane proteins are dispersed and individually inserted into the phospholipid bilayer. Hydrophobic regions would be contained inside the core of the membrane, which is a non-aqueous hydrophobic environment. So again, that's kind of a simplified version. Here's a more complex picture of the membrane. Okay, we say that the membrane is a mosaic of components. Phospholipids, cholesterol, proteins, and carbohydrates. We say that it's fluid because these components are not static. They're not locked in place. They're able to move around in that membrane. And so lipid molecules will move to the left, to the right, forward and backwards. They may even flip-flop. They may go from the exterior of the membrane to the interior of the membrane. Right? And protein molecules, same thing, they can move around. Okay? So that's the model that's still accepted today, the fluid mosaic model. Let me just show you a little more evidence, just one more piece of evidence to support this model. So two scientists by the name of Fry and Edidin, they did an experiment to actually show that the membrane is fluid. Okay, and so they took two different cells, a mouse cell and a human cell, and they labeled the proteins in each of these cells. And so in the mouse cell, they labeled all the proteins with in this case, it looks like a pink fluorescent dye. In the human cell, they labeled all proteins with a purple or blue, whatever this color is, fluorescent dye. So two different fluorescent dyes. They then created a hybrid cell. So they allowed these two cells to fuse. Okay, and they had these cells in a petri dish, right? And they waited about an hour after that fusion, and they went back and they looked, and they were looking at the distribution of the fluorescent markers. And what they found was that, they had moved. If they hadn't moved, then there would have been a line straight down this cell with all the pink proteins on one side and all the purple proteins on the other. But that's not what they found. They found that the proteins had moved. Okay, and so again, that's good evidence that the membrane is fluid. Okay, so let's remember again what a phospholipid looks like. Just want to make sure you have this. And so phospholipids, you're familiar now with this little picture, right? We said that this is the hydrophilic head group. And here we have our two hydrophobic fatty acid tails. And so the same thing as over here, just a different picture, a more complex picture actually showing the atoms involved here. And so a phospholipid is going to be composed of glycerol. So this is glycerol right here. Okay, which the end carbon here is going to be covalently bonded to a phosphate group, which carries a charge, which makes it polar and hydrophilic. These two carbons are going to be attached to fatty acids. And so here's one fatty acid. This is a straight chain saturated fatty acid, all single bonds between these carbons. And this second fatty acid tail happens to have a double bond, which makes it unsaturated and bent. Notice the shape there. We'll come back to that. These two tails are just made up of carbon and hydrogen, which are nonpolar, right? And so these tails will be hydrophobic. And so we call a molecule like a phospholipid, we say again that they are amphipathic, just like proteins. They're going to have two distinct regions, one polar and one nonpolar, or we can say one hydrophilic region and the other region would be hydrophobic. Because of that, if I were to take just a handful of phospholipids and throw them in water, they would naturally form one of these structures, okay? So right here I'm showing you a sphere, a bilayer of those phospholipids with a hollowed out internal core, okay? Which if I were to just take a piece of this, it's the phospholipid bilayer, okay? And sometimes lipids will form a single layer sphere as well, depending on those lipids that are in there. Okay, but again, membranes are going to look like this, a phospholipid bilayer. And they'll form, a spherical structure, and the inside would be the inner contents of our cells. Okay, so again, phospholipids have a natural tendency to arrange themselves in a bilayer when put into an aqueous solution. The hydrophobic tails are going to face each other, where those hydrophilic polar phosphate-containing head groups will be facing the outside aqueous environment as well as the inside cytoplasmic environment. Okay, how about proteins? So again, proteins are going to be individually inserted into the membrane, and they will be able to move around in the membrane. There's a couple different types, there's lots of types of membrane proteins, but a couple different classes that we put membrane proteins into. And I've mentioned this already a few slides ago, so let's make sure we have it. So proteins can be integral or they can be peripheral. Those are really the two main categories. So what does the term integral mean? Integrated into the phospholipid bilayer. So they're within either partially or fully within that membrane. Okay? They can be transmembrane, which means that they span all the way from one side of the membrane all the way to the other. And so actually all the examples of integral proteins on this figure are transmembrane. So here's one. So this first one right here is a transmembrane protein. It starts in the cytoplasm, it goes through some alpha helices here and comes out the other side. Okay? This region of the protein here, as well as this region of the protein here, would be the hydrophilic regions. And this region in here tucked in the membrane would be the hydrophobic region of that protein. This is also a transmembrane protein. Look at it's going to span 1, 2, 3 times. Okay? And so again, the hydrophilic regions will be in contact with the cytoplasm and the outside of the cell, where the hydrophobic regions will be embedded into the core. Here's another transmembrane protein. This is a barrel protein. This is probably a channel protein. It's involved in transport. Again, transmembrane goes all the way from one side to the other. Another example of a transmembrane protein, but here it's attached to this. This is a peripheral protein. And so what's a peripheral protein? These are going to be proteins that are going to be found either on the exterior or interior surface of the cell. And they can be either associated with a protein or they can be associated with these lipids. So in this case, this one's an external peripheral protein associated with this integral membrane protein. In this example here, we have another peripheral protein. And again, it's associated with the cytoplasmic side of an integral protein. Okay? So you want to make sure you understand the difference between integral and peripheral, and you want to know what a transmembrane protein is. Not all integral proteins span the whole membrane, but all transmembrane proteins do. Okay? The last component, almost last component. So we've got lipids, we've got proteins, and now I want to talk about carbohydrates, and lastly we have cholesterol that we'll talk about. So on this slide I'm showing you carbohydrates. So carbohydrates can either be attached to lipids or proteins. And so if the carbohydrate is attached to a lipid, like it is right here, so these little green structures, these are individual glucose molecules, right, forming a carbohydrate. If it's attached to a lipid molecule, it's called a glycolipid. So glyco means carbohydrate. If it's attached to a protein, like here, it's called a glycoprotein. So we've got glycolipids and we've got glycoproteins. Okay? Carbohydrates are only going to be attached on the external membrane surface. We're not going to find them on the cytoplasmic side. And these carbohydrates can be straight chain or they can be branched. Okay? They're involved in things like cellular recognition. So again, your immune system is responsible for making sure that everything in your body belongs to you. And so the surface of your cells are going to have markers that tell your immune system that those cells belong to you. As they survey different cells in your body, if they come across a cell that does not contain those surface markers, then they know that that cell doesn't belong to you. They would consider that to be non-self, and they would target that cell, whatever it is, a bacteria or whatever, for destruction. Okay, so your surface markers are really important in immunity. Collectively, all of the carbohydrates found on your surface is called the glycocalyx, which literally means sugar coating. Okay, and so these carbohydrates, again, multiple functions. They are also highly hydrophilic, and so they will pull, water towards the surface of your cells, and with that water will come ions and nutrients, and so it's important for your cell to have that sugar coating. The last slide. Here is the last slide, and there's no room for me, maybe, right there. Okay. we're going to talk about membrane fluidity. And so this is when we're going to bring in cholesterol, which is the last component of membranes. But before we do that, I want to talk about membrane fluidity, kind of ignoring cholesterol for a minute. Not all cells have cholesterol. Cholesterol is only found in animal cells. So when we're talking about plants, for example, there is no cholesterol. So again, two factors are going to determine the degree of fluidity of our membranes, how fluid they are. You don't want your membrane to be too fluid and fall apart, and you don't want your membrane to be too rigid and almost freeze. You want there to be this medium level of fluidity, so things can move around, but things don't fall apart, okay? So the first factor that is involved in determining membrane fluidity is the actual phospholipids that are present. And the second is cholesterol. Okay, so let's look at the phospholipids first. So you've seen these terms back in chapter 3, unsaturated versus saturated, right? And so this first membrane that I'm showing you here, has a lot of unsaturated fatty acid tails. So all these kinks that you see are because of double bonds. Okay, and the more kinks that you have, the more space there is between phospholipid molecules, and that makes the membrane more fluid. The more saturated fatty acid tails are present, the less kinks, the less spaces. And so we would say that this membrane here is more viscous, more rigid, okay? There are animals that will actually, or organisms I should say, some animals, that will actually change the composition of their fatty acid tails in response to temperature changes. So I gave you one example here, fish living in aquatic environments. Their response to a decrease in temperature, so their response to colder temperature, is actually to change fatty acid composition. So if their membranes had a lot of saturated fats, as it gets cold, these can pack really closely together, and it could again make that membrane too rigid. And so in colder temperatures, what will happen is their membranes will change composition to create more double bonds, which gives the membrane more fluidity. Okay. And then in some animals, cholesterol does that job, kind of for us. And so what is cholesterol? We consider cholesterol to be a fluidity buffer. So again, cholesterol is going to be a spacer molecule that's going to make sure our membranes don't become too viscous, too rigid, and also not too fluid. Okay, so we say it's a fluidity buffer. So this is what cholesterol looks like. It's embedded in the membrane. It's a hydrophobic molecule, right? It's a hydrophobic lipid. Again, cholesterol present in these membranes is going to reduce membrane fluidity at higher temperatures, but at lower temperatures, it hinders solidification. So it keeps the membrane fluid when it gets cold, and it prevents the membrane from becoming too fluid when it's warm. Okay? That's the end of part 1.